Howdy, this is Browdy. This is the third and final part of the cell membrane and transport notes. And so far we did the very first lecture about lipids and polarity. Then we talked about cell membrane and transport part one, which was kind of just like the structure of the cell membrane as well as kind of the main types of cell transport. Then part two was all about passive transport, the different types, osmosis, solutions. And we talked briefly about active transport. And today, part three is focused entirely on active transport. So we ended last time talking about how active transport is the type of transport that requires energy to go from low to high concentration. But there are kind of other types of active transport when bigger molecules are involved. So the first thing that we're going to talk about today is something called endocytosis. So endo is a prefix that means in. We saw that with endosymbiosis, right, which is the theory about how chloroplasts and mitochondria ended up becoming the precursors to the initial eukaryote as they were engulfed by another cell. And so endo means in, cyto, we've said in the past, means cell. So endocytosis is the process of bringing large molecules into the cell. And so specifically this happens using vesicles and using energy. So if you remember in Unit 2 we talked about how vesicles are kind of these bubbles that form either off the side of the Golgi apparatus or other things or from the membrane. And when we did the bubble lab, we talked about how when you blow a bubble, essentially you're forcing air into the membrane and eventually it pinches off and then everything reforms. And that's kind of what's happening here. So in endocytosis, we've got these larger molecules that are too big to just be brought across the membrane using active transport and protein pumps. These are too big for that. And so these molecules are coming up and they're hitting the side of the cell membrane and they're kind of denting it a little bit. And eventually they're putting so much pressure that this bubble, this vesicle, kind of pinches off and the membrane itself reforms, and then the bubble can move into the cell. So that's endocytosis, the process of bringing large molecules into the cell. Now we'll talk more about this. There's also a special kind of endocytosis called phagocytosis. Phag or phage means eat. And so this is specifically the type of active transport that happened when this chloroplast and mitochondria were engulfed by another cell. So phagocytosis is where a cell engulfs a molecule. So essentially it kind of spreads out around it, forms a circle around it, and creates a vesicle simply by surrounding and engulfing the cell. So normal endocytosis, something kind of comes up against the cell membrane and then pushes and makes a bubble. Whereas phagocytosis, the cell is actually engulfing the thing. The next kind, or the opposite, is called exocytosis. And so if exo means out, you could expect that exocytosis is the process of expelling material from the cell or moving things out of the cell. And so here we have just a very simple picture that shows both. So extracellular fluid means it's the stuff outside of the cell. And if here's the cell membrane, and we'll say the yellow is the cytoplasm, so the inside of the cell. Here we see endocytosis. So something is coming up to the cell, and these molecules are hitting it, and they're forming a little dent. It almost looks like a pore. And eventually, when these come up here, it's going to kind of pinch off, just like if you're blowing a bubble. And that bubble then is containing the molecules and it moves into the cell. The opposite is if a vesicle, say there's a protein in it and it was sent from the Golgi apparatus or something. It could be going up, hits the cell membrane. They fuse and this opening forms, and then the insides get expelled from the cell. They get sent out from the cell. And that's really what endo and exocytosis are. So here, just looking a little more step by step and slightly more detailed, here we see the cell membrane. So if here's the extracellular fluid or the outside of the cell, we'll say this is the cytoplasm or the inside of the cell. Here we see the phospholipid bilayer, right? The polar heads, the nonpolar tails, and something is coming up to the cell and it's sort of making a dent, right? And eventually this is going to make a big enough dent that these fuse and it pinches off. And when that pinches off, now we have this membrane that's reformed and then a phospholipid bilayer around the molecule, right? That's a vesicle. It's literally just a membrane. So it's made up of the same thing. And that goes in, and eventually when it gets where it needs to go, it breaks apart. The membrane kind of refuses into the things around it, and it can go where it needs to go. Exocytosis, on the other hand, here we have a vesicle, so we could say that this is from the Golgi apparatus, and there's a protein, or in this case, it looks maybe like a lysosome, right, which is getting rid of waste. It's a vesicle that is sending waste out of the cell. And so maybe this is a lysosome that's trying to get rid of waste. In any case, it's going towards the cell membrane. It makes contact, and then we can see kind of there's a little bit of pressure here, and as that pressure increases, these membranes fuse, and this opens up and expels everything from the cell. And that's endo and exocytosis. It's pretty straightforward. Endo is in, exo is out. Phagocytosis is a special type of endocytosis, but really, those are the forms of active transport that are most important to know. Now, there's one other part, there we go, of the notes, and I apologize for clearing my throat. There's one other part of the notes, and the very end here just wants you to remind yourself of the main parts of the cell membrane. We have 4 main parts that we talk about, and I'm going to talk about them in a little bit more detail here. So, the very first thing we've got are things that I like to compare to Q-tips, the things that are really the main structure, and those are the phospholipids down here. So this shows the whole width, the phospholipid bilayer, the phospholipids, you can call it whatever you would like. The next thing that we have are these here, and these are proteins or protein channels or transport proteins or carrier proteins. You could really hear it called any of those things, and the idea is the same, right? It gets a molecule from one side to the other, regardless of the direction. Protein channels or protein pumps, transport proteins, carrier proteins, all are really the same idea. There's cholesterol, which strengthens the membrane. It makes it a little thicker, a little tougher. It also keeps the lipids from sticking to themselves, right? So that's cholesterol. And then lastly, there are glycoproteins here. And these are kind of what receive signals from the cell. They are what identify the cell to other cells. These could also be called carbohydrate chains. So glyco means sugar. So these are kind of combinations of protein and sugars are carbohydrates. So if you see this called a carbohydrate chain, it really is the same idea. So those are our 4 main components: phospholipids, protein channels, cholesterol, and glycoproteins. And that's it for the cell membranes. The entire unit is covered in these 4 lectures with these 4 different parts. So I hope this has been helpful. I'm going to try to keep it short and sweet, and I will see you in class. Have a good one.